Modifications on your chromatin can be passed on to future generations. We call that epigenetic inheritance. Now, unlike a DNA mutation, remember where I changed the letters around, these changes to chromatin can be reversed. We can demethylate DNA, so we can loosen and we can tighten that structure. This is going to explain the difference between identical twins. So because of this varying tightness versus untightness of that DNA packed together, this is going to influence gene silencing, where I completely turn off certain genes from doing anything. Histone acetylation was that whole unwinding process to allow transcription to occur to read those genes and make proteins. X chromosome inactivation, silent chromatin. So you can pass on this chromatin structure to your children, and this also explains why two identical twins might have slight variations in their face or their height or their hair. Things can get passed on in those regards. Here's a video you should watch that'll help you out. Now, transcription initiation. Remember, this is going from DNA to RNA. Now, specific transcription factors can bind to your genes and enhance or slow down the amount of expression that's going to occur. So we kind of went through this with the operons. Activators are elements that increase transcription. Repressors are going to decrease transcription. Just to review again, because I feel like I'm losing some of you and I can't even see you. DNA to RNA to protein. Here is my transcription part. Here is my translation. I am focusing on this whole step right here, transcription. How much RNA, how much reading is actually occurring in my cell. Transcription initiation complex. So this is my start of transcription. Again, you can see that activator. So activators are going to bind to enhancer regions. We're going to increase the amount of transcription. Now, cells have specific transcription things that need to occur. So some genes need to be turned on for one cell, but they don't need to be turned on for another cell because they're not going to use that. One example that I put in here is the difference between a liver cell and a lens cell. So lens cell, what do you think that belongs to? Eyeball. So let's take a look. Same DNA, same genes, but the difference is what is turned on versus what's turned off. So if I look at this liver cell on the left, albumin gene needs to be expressed because it's needed. However, the crystalline gene is not expressed because it is not needed by the liver cell. The gene is there, but it's not being expressed. When I look at the lens cell, albumin gene is not expressed because it's not needed. But the crystalline gene is going to be expressed because it does need it. So while you might have similar DNA in cells, the difference is what is being expressed, what is actively being shown. By shown, I don't mean you can visibly see it. I just mean what is being expressed, what is being transcribed. Now there's some regulation that needs to occur with RNA when it comes to that whole activation process. We have microRNAs or miRNAs, and we have small interfering RNAs. And they can bind to mRNA and degrade it or block translation. So they can stop the protein from being made. Stop protein from being made. So let's look at our little diagram. We have mRNA in the cytoplasm. Again, it needs to get to the ribosome in order to make a protein. Now here's the translation, and over here you can see normal steps. Translation to polypeptide, the protein is processed, it's active, it's going to be transported to its particular location. However, sometimes I might need to degrade it So I'm going to have microRNAs or siRNAs come in and degrade either the mRNA or that protein or the stuff that is not needed. There's a little bit of a better picture of that whole process that I just talked about. Here's a nice video for you to watch. Here's a quick summary of pictures and little blurb boxes for gene expression, chromatin modification. And now let's get into embryonic development. Embryonic development. So this is where we're going to get into a little bit of a stem cell conversation and why things develop the way that they do. When we're going to talk about zygote to organism. So zygote to organism, and there's some steps in between. You got zygote to embryo to fetus to organism. Zygote is that collection of cells, so just when fertilization happens. So there are a few ways that development is going to occur. First is cell division. So large number of identical cells through mitosis. So one cell turns into 2 cells, turns into 4 cells, turns into 8 cells, turns on to more cells, dot to dot. We're going to go on forever and ever and ever. So many cells later, we have some development occurring. Cell differentiation is when cells become specialized in structure and function. So this would be my eye versus liver cells. So cells are going to start to have particular jobs and roles in the organism. And then you have morphogenesis. This is when cells are going to aggregate together in certain spaces to determine left from right, top to bottom, tail to head. So aggregation of cells in certain places. We're going to talk about some of the proteins that determine where the top is versus the bottom. So cell determination is an irreversible series of events that leads to cell differentiation or the specific job that a cell has. And we talk about stem cells here. I'm going to use some different colors because I'm getting bored with that color. So over in this diagram, I have a lot of stuff going on. I have some different phases. So here's an oocyte. That's how I like to call it. Here is a sperm coming into contact with the egg. This is when fertilization has occurred. And development of cells is going to start to happen. And I have some totipotent cells here. A totipotent stem cell is capable of becoming any cell type, or it can become a complete embryo. So this can become any cell type, or it can become a complete embryo that can turn into an eye cell, a liver cell, a brain cell. It can turn into anything, or it can turn into an embryo. So here's that whole kind of process occurring from fertilization to totipotent to blastocyst to a human fetus. But we also have pluripotent cells. Pluripotent cells are capable of becoming several different cell types. So they can turn into a cell in the circulatory system. They could turn into a cell for the nervous system. They can turn into a cell for the immune system. They can turn into any kind of cell beyond this fetus cell. And then you have unipotent cells. They can only become one cell type. They're limited in what they are allowed to turn into. You have cytoplasmic determinants. These are substances in the egg that are distributed unevenly in early cells of embryo. And if I look at this picture, I have molecules of 2 different determinants. I've got these triangles, and I've also got these orange circles. And if you look at the steps that are occurring in this diagram, you can see that all of the orange circles migrated to the left, and all of the green triangles migrated to the right. And this is going to dictate that cell division process. Cell divides, and most of my triangles are in one spot, and most of my circles are in another spot. So I'm determining some features about this cell. Induction is when cells are triggered to differentiate. They are triggered to turn it into something. Now remember, cells talk to each other and they know when they're close together. So they are going to be forced to turn it into something. You also have cell-to-cell signaling. Remember, cells talk to each other. These cells are going to be triggered into becoming some sort of growth factor or influence other neighboring cells. So kind of the big picture here is cells talk to each other and help dictate what each other will become. So let's take a look at this cell. Top left, embryonic precursor cell. This is the beginner basic cell. Has nothing special about it. Has no job yet. It's just a cell. Don't know what kind yet. So if you look, this regulatory gene, MyoD, is off and muscle-specific genes are turned off. So right now this is a cell with no job, no function, no name, just a precursor. It's a building block. Well, some sort of event will occur and now it has been determined to become a myoblast, which is it's related to the muscular system. So now this regulatory gene has turned on. However, our muscle-specific genes are still off. mRNA is going to start to be transcribed, and now we have turned into a fully decided muscle cell. So this precursor, through a series of regulatory steps, has now been put into that muscle cell category. All of my muscle proteins are turned on. All of my transcription factors related to those muscle proteins are on. This will now turn into a muscle cell. Pattern formation. So this is how we set up the body plan. So head, tail, left, right, back, front. So cells determine body structure during development, during that cell differentiation process. So here's my egg developing. Here's the unfertilized egg. Here's the egg that has now been fertilized. Here is when it starts to become a segmented embryo. So here is where the head will develop. My thorax, well, not mine because I'm not a fly, I think. And here's the abdomen. So pattern formation is going to start to occur, determining what is up, down, right, left, butt end, head end. Of course, we don't call them those names. Top would be dorsal, like a dorsal fin of a shark. Ventral is down, so belly. Anterior is the head side. Posterior is the tail end side, and then you have left from right. Morphogenesis. Substances establish an embryo's axes, so they determine what is left, right, up, down, and one of those particular substances is called bicoid. So bicoid, when it aggregates together, is going to determine the anterior side or the head side. So when lots of bicoid is at one end, that is going to allow us to determine this is what will develop into the head. So then the opposite would be the posterior. And then we can establish through a series of good guesses where our dorsal side will be, where our ventral side will be. Homeotic genes are the controlled genes that control pattern formation, so where things should be developing on an organism. So we have wild type versus mutant flies here. Wild type is normal. Those Hox genes determine where the eye should be, where the antenna should be. And here's my mutant, and we have a little problem here. We are now growing legs where the antenna should be. So there's an issue with the Hox genes. Those Hox genes control the pattern formation where things are supposed to develop. Here's a great video to watch as well. The PITX1 gene is an example of a homeotic gene. Homeotic and Hox genes are the same things. We just got tired of writing out homeotic. So the PITX1 gene has instructions to make a protein that is critical for the development of the lower limbs. So in stickleback fish, it is responsible for the development of these pelvic bones. So that would be the lower limb of that fish. In humans, it is related to development of anterior structure, the brain, the structure of hind limb. Just like we said, those lower limbs, when they're mutated, they may cause clubfoot, polydactyly, extra fingers and toes, upper limb deformities. So this particular Hox gene is responsible for a protein that's critical in the development of hind legs. Apoptosis plays a role in organism development. Remember, apoptosis is everyone's favorite word because it's programmed cell death or programmed cell suicide. Most embryonic cells are produced in excess, so we produce more than we need. So cells go through apoptosis to sculpture some of those organs and tissues. These are going to be carried out by caspase proteins. Caspase proteins. So if we take a look at this little foot here, we've got too many cells. So some cell death is going to start to occur. We're going to start to form space between those digits. So we killed off those excess cells that were not needed. All right, back in Unit 5, when we talked about cell division, we talked about proto-oncogenes. So we're going to talk about control of the cell cycle one more time because this is related to gene expression. So onco, we think of oncologist or oncogenes, and they usually mean cancerous. So proto-oncogenes stimulate cell division. So they make cell division happen. Tumor suppressor genes are going to inhibit cell division. They're going to stop it. They're going to block it. Now, when there's a mutation that occurs in these genes and I overexpress, then I'm going to have too much cell division occur. And that's what leads to that cancer. It's going to keep going. Nothing is there to put it in check and stop it. So proto-oncogenes, they're genes that stimulate that normal cell growth and division. Now, oncogene is a mutation that's going to occur. It's going to have this cancer-causing effect. It's going to increase the product of proto-oncogenes. So I'm going to increase the amount of cells. I'm going to increase activity in those cells. So the cells are going to be overworking and doing jobs that I don't want them to be doing. Here's a quick flow chart of proto-oncogene to oncogene and all of the things that can occur. We talked about RAS and P53. Again, I'm not really going into full detail because we went over this in Unit 5. So the RAS gene is going to stimulate the cell cycle. It's a proto-oncogene. When it's mutated, and this tends to happen in 30% of cancers, we have permanently activated this RAS gene. So it's going to cause the cell to divide constantly. When a mutation occurs in the RAS gene, this normal RAS gene, it will cause it to be permanently activated and it's going to divide constantly. The P53 gene is a tumor suppressor gene. It's supposed to stop the cell cycle, initiate DNA repair, activate apoptosis when necessary. When it's mutated, and it's usually mutated in about 50% or more of cancers, we have stopped DNA repair from happening and we stop the cell's ability to commit apoptosis. So those cells will just keep dividing and they won't be killed off. We also in Unit 7 talked about the 7 different changes in DNA that occur in order to cause a cell to become cancerous. And we also talked about how the longer we live, the more accumulation of mutations that will occur and the increased likelihood of a cancer occurring. So I hope this helps. Please read the chapter, look through your notes, watch Khan Academy. If you have any questions, please let me know, and have a great weekend.